So uh, today let us talk about some questions related to cystic echinococcus or hydatid disease. So first of all, what is the causative agent of cystic echinococcus? The answer is Echinococcus granulosus. Next question is, how many morphological forms does it have? The answer is 3. Adult worm, which resides in the dog's intestine, larva, which is also called as hydatid cyst, and eggs, which is the infective form. Next question, what is the name of the larva? I already told, it is hydatid cyst. Which one is the pathogenic form? The pathogenic form is the larval form. I mean, which one? What does this question mean? Pathogenic form, what does it mean? It means which form causes the disease, this hydatid disease. That is the larval form. That is hydatid cyst. Which one is the infective form? Which one infects the human with who is an accidental host? That is the eggs. The cyst, this hydatid cyst, the diagram of the cyst is important. This, how many? The question is, how many layers does the cyst wall have? It has 3 layers: periocyst, ectocyst, and endocyst. Periocyst is host derived, ectocyst is laminated membrane, and endocyst is the germinal layer. This structure of the cyst, if you see the diagram, it has a periocyst, ectocyst, and endocyst, and inside it has daughter cysts. It has brood capsules containing the scolex, protoscoleces. So these brood capsules which contain the protoscoleces, which of the layers give rise to this brood capsule? The answer is endocyst. What does the brood capsule contain? The answer is protoscoleces. These protoscoleces can give rise to new hydatid cells. Is hydatid fluid antigenic, toxic, and anaphylactic? The answer is yes. That's why if the hydatid cyst ruptures, there could be anaphylactic shocks. What is hydatid sand? These brood capsules or protoscoleces break off from the endocyst and get deposited at the bottom of the cyst as sand-like granules. That is known as hydatid sand. Fate of hydatid cyst. There could be spontaneous resolution or calcification, or there could be rupture. If rupture occurs, there could be 2 things. One is secondary hydatidosis, formation of daughter cysts, and another is anaphylactic reaction. I just told that this hydatid fluid is anaphylactic, antigenic, and toxic in nature, so can cause anaphylactic reaction. That is very serious. Laboratory diagnosis, which is the main part of microbiology. So the first will be demonstration of hydatid cyst or cyst components in imaging. So the fluid aspirated from a surgically removed hydatid cyst can be subjected to direct mount or staining with acid-fast staining to detect brood capsules and protoscoleces. There could be histopathological examination. Surgically removed cysts can be subjected to histopathological stains like H&E to demonstrate the 3 layers of the cyst wall and the attached brood capsules. Serological tests are antibody detection tests like ELISA using the B2T antigen and or DIG-FA, that is dot immunogold filtration assay. The confirmatory test is Western blot, which is the most specific serological method. It detects antibodies to hydatid cyst, fluid antigen, or antigen B fragment. Imaging methods could be used like X-ray, USG, CT scan, MRI. If you're asked what is the imaging method of choice for the diagnosis of hydatid disease, that is USG. This is another MCQ. USG helps in determining the exact location of the cyst, the size of the cyst, how many cysts are there, number of the cysts, or activity. That is, whether it is active or dormant. USG is also used to monitor the response to treatment, that is prognostic use is also there. Now there is another question, that is what is water lily sign? That is question number 16. It is found in the USG. The membranes may be detached and floating within a cyst cavity. The membranes of the hydatid cyst can be detached when these membranes gets detached and floats in the hydatid fluid, resembling a water lily. That is water lily sign. MRI can also be used as an imaging method. Molecular methods are also there. PCR targeting mitochondrial DNA. Cassoni test is obsolete now, but it can be asked in MCQs that it can demonstrate an immediate hypersensitivity reaction, this type 1 hypersensitivity reaction that develops following injection of hydatid fluid antigens. The question number 17, you can see about hydatid, this Cassoni test. But it is obsolete, it is not done to diagnose hydatid disease. These are also MCQs. Which antigen is used in ELISA? B2T antigen, recombinant antigen, to detect antibodies. What is DIG-FA? The full form can be asked. Dot immunogold filtration assay. It is the full form. Which test is used for confirmation? There is Western blot or immunoblot. So there is a summary table given here. for revision. So this was all about the questions on hydatid disease.